the nutrients diffuse out of these digestive ceca into the coelomic fluid, and the way those nutrients get distributed to the cells, the tissues of the body, is this coelomic fluid in here, just bulk movement, bulk mixing of that. There's no specific heart. There are some cilia here that line the raised parts that create a little current. But the other thing is, we'll see when we talk about the water vascular system momentarily, there are these structures called ampulla, and they're inside here. And these things move around a bit. So the movement of these structures move the bulk fluid, the coelomic fluid, distributing those nutrients, okay? So they're called ceca because they're outpopping, but they're very different in terms of overall, okay? Any other questions? Yes? So there are ceca in each arm. So this has radial symmetry, usually pentameric, but some have 10 arms, some have 8, but 5 is the most common. But yes, these structures are repeated with each arm. What would be a term for that? Metamerism, okay? So yes, so what they've done here is they don't show you the digestive ceca here so that you can see the gonads oral relative to those structures, because otherwise they would hide them, okay? So what they've done here is in this drawing, here they have the digestive gland not showing structures underneath, here they remove the digestive gland to allow you to see the gonads, then here they've also removed the gonads, first they remove the digestive ceca, then they remove the gonads, and then you can see clearly the ampulla, okay? So they've done sort of some things here to make a specific focus on each arm, okay? So we have, most digestion occur here, we have waste and additional processing fluid occur, so it can go out from this pyloric stomach, outside the digestive ceca, then out through a very short intestine. And it's really hard to see this intestine, so let me just draw you what it kind of looks like. And I apologize because this is fairly confusing. So they have an opening, the mouth, on the oral surface, and then just do boxes. They have the cardiac stomach, and they evert that to feed. They literally stick it outside their body. So if they're trying to feed on a clam, the clam tries to close its shells. What muscles would it use to close its shells? Adductor muscle. So the clam uses the adductor muscle. The starfish grabs on, uses those tube feet, which are sucker-like devices, generate a fair amount of force for each tube foot, and they basically try to pull it open. And what they're trying to do is separate that mantle cavity, the mantle, so slightly so they can evert their stomach inside that mantle cavity. And once they have their stomach inside that mantle edge, they start releasing digestive enzymes, and the stomach goes in and grabs the food. It then gets transferred to the pyloric stomach. And this is oral, going to be ab oral on this surface, okay? So this is just like kind of a section through like this. So food processing, I don't have any colored chalk, unfortunately. So the food's going like this. Then it goes out to the pyloric ceca. All right? Processing occurs here. Nutrients diffuse here. Ammonia, et cetera, things like that is produced, gets into the coelomic fluid, and then it can diffuse outside the body. But then we have additional processing. So once we processed in here, it comes back through here, then through a short intestine, out an opening on the ab oral surface. And this opening is called the anus. But before we do that, we have some additional ceca here that are called rectal ceca that do additional processing of the food. Okay? So it comes back here, and then processing here, and then eventually wastes out here. We saw sort of a similar pathway to some extent in the crayfish in terms of the stomachs. They had a mouth, they had an esophagus, they then had a two-chambered stomach, which is the cardiac here, closest to the mouth, and pyloric, and within here was the gastric mill, we'll see this shortly, that fragments food. And then food that gets here is small enough to be digested, and then it goes through the intestine. Then out the anus. So I'll show you that pathway shortly. So this concept of two stomachs, if you look at the rat stomach carefully, how many of you notice that there are two very different looking compartments of the stomach? You notice that? So if you look at the rat, two portions of the stomach, they look very different color in that, they have kind of different functions. But they usually, we don't go over the two names of it. So anyway, so that's the digestive system. Nutrients diffuse out. In terms of movement, we have a water vascular system, which we'll show shortly here. Again, realize they just remove structures to help you see things. This is the aboral surface. They show this structure called the major porite. That's where it's believed water enters into the water vascular system, which I'm going to go over now. So this figure here. So it's believed that fluid enters the water vascular system through this major porite. And remember the orientation. The orientation is oral on this surface, aboral here. So we're going to work, in this case, from the oral surface towards the oral in terms of the pathway. Fluid enters here, then there's this structure called the stone canal. It leads from the major porite to the ring canal. And if you want, just think of it as a ring that you put your finger around. That would be the ring canal here. And then it runs out in the radius here of each arm. So that portion of the canal is called the radial canal. Then it runs laterally within each arm to the sides, and those portions are called lateral canals. And then they lead to these 2 foot on the oral surface, exposed to the outside, and the ampulla inside. So here shows you a cross-section here. So this is cross-section of the arm. And what we see here is this radial canal, that line there. We see the lateral canal that leads to each of the ampulla here. There's the ampulla, there's the 2 feet. And this is how we can see the coelomic fluid gets moved around, because in order to move these 2 feet, there's muscles that contract in the ampulla move around a little bit, and that would be bulk mixing of the fluid here, okay? And if you notice this cross-section, the digestive ceca are above, that is aboral, and the gonads are oral relative to the arm. They do extend out in the arm, okay? That's the water vascular system. They use these 2 feet to hold on, generate force, etc. And there's actually some interesting collagen here. We know that collagen is a unique protein found in animals. No other organisms have it. And they have a special form of collagen called catch collagen. And so the clam typically doesn't have much of a chance, because what happens is when the starfish grabs onto the clam, it actually modifies its catch collagen so that it can exert a force, because of the way the collagen is designed, without really expending ATP. So they generate the force by modifying their collagen, whereas the clam always has to continually expend a force and ATP to overcome that catch collagen. So the starfish can just hang on for a long time, generating that force without lots of expenditure of energy. So usually the starfish wins. Not always, but usually. Okay? So there's some work being done about that collagen because it's so unique. In terms of damage to tissues in that, a lot of engineering of tissues, you can now use 3D printers to print a scaffold, a matrix, that you can implant stem cells in and try to regenerate tissues, potentially maybe entire organs for humans. So a lot of work being done on that. This shows you, again, just a little bit better drawing. This shows you the digestive ceca. Here you can see how highly branched it is. So this is the pyloric ceca, pyloric stomach. This is the cardiac stomach underneath. Okay, I'll show you some images. And there's the branches that lead out and also back. That intestine's short, so you never really see that. You will see these rectal ceca, though. You'll see those branches. You don't typically see the intestine. So the branches you can clearly see will be the cardiac stomach, the pyloric ceca, and these rectal ceca, but not really the intestine, okay? And here shows you these 2 feet. And at the ends here, they're somewhat modified. They actually detect light. They're not image form, but they can detect light. So over a long weekend, if I go into the cold room and turn the lights on, a lot of times the starfish will kind of jerk because it's a sudden light exposure that wasn't there before. Here's the gonads here. These are dioecious, meaning they have 2 different genders, males and females. The gonads in the males will be a whitish color. and the females kind of an orange or salmon color. Okay? If they're reproductively mature. Okay? Gonads vary in size. If it's not reproductive season, they'll be quite small. Question. So the question is, does cardiac stomach have anything to do with cardiac muscle? No. Okay. The term cardiac does mean heart-like, but you wouldn't find cardiac muscle within the cardiac stomach. Okay. And I'm not quite sure why they gave it this name, cardiac and pyloric. I don't know the historical derivation of that, but that's the terms they use. Okay. I can look into that and see if I can try to figure out the history of why those terms are used. In terms of the structures we see on the surface, here's those spines. So those are just the modified endoskeleton that project upward. Here's the coelomic lining projecting upward out past the endoskeleton. So those are those dermal branchia for gas exchange. We have these modified structures here called pedicellary, the little pincher-like things. There's one open, the majority of them are closed. In lab, the instructor will take and use a TV monitor system. We'll have a cut piece of the arm. We'll take and have them rip a piece of paper towel, so you get little strands of paper, and you can stick it in there, and then you can watch microscopically. You can see these pedicellary open and grab onto the little strands of paper. In the tank, we're going to have the instructor reach their arm in. Hopefully, the hairiest one of them will reach their arm in, and they'll go next to the starfish. And the starfish will grab the hair with their pedicellary, and then they can pull their arm, hold the starfish up, and you'll see their hair being pulled. You can see their skin puckering because those pedicellary have grabbed on. So they help remove ectoparasites. So it's a defensive mechanism to help clean the surface. Here shows the major porite. Here's these very spines here. So if you touch this, you would feel that it's quite hard. But there's actually endothelium on top. Sorry, there's this layer of epithelium on top of this, and you can certainly feel this where the spines aren't projecting up so much. Okay, and the leather starts real obvious. Here's a side view showing you the spines here. Here's these 2 feet. The spines are somewhat modified on the oral surface to protect these 2 feet. Here's that leather scar. And the cool thing about these, they clump their dermal branchia together. They're actually large enough you can pretty much see them with the naked eye. And these are those branches of the dermal branchia. But again, since we have this displayed on the monitor system, you'll see their dermal branchia, you'll see those pedicellary, and you'll see those spines, how big they are. Okay. This is looking from the oral surface. This is the mouth. Here's these 2 feet. Here's those spines. Notice how they're modified to protect these 2 feet. This is sometimes called the ambulacral groove, where you have these 2 feet and modified spines. This is reproductive season. Huge gonads. This is a female. Sometimes in our marine tanks we have enough organisms that the water quality gets affected a bit, and these things will actually burst because the gonads get so large. Now in nature that's probably not the way they really work. Okay, but in terms of that, there is some asexual reproduction in starfish, certain species. So it used to be that fishermen who were trying to harvest oysters and clams and things like that, they knew the starfish were competing for them. So what they would do when they get starfish, they'd cut them up into chunks and throw them back into the sea and have a hearty laugh. But actually the starfish just regenerate themselves. So they were actually increasing the population density because they promoted asexual reproduction. So I guess even if your goal is to kill something, you should know more about the biology of it, okay? Because you may not be doing what you think you're doing, and a lot of times we've introduced organisms to control one species, and we created way more of a problem than we had before. So you really need to know a lot of biology if you start experimenting with a lot of things. Here's a male. The gonads here are white and colored. Now this is called the ambulacral ridge. This is that endoskeleton internal, and the ampullae aren't too visible here, but they're off to the sides. Those pipette-like bulb-like structures, okay? And then this just is a diagram again. Here you see those ampullae, the 2 feet here, the ambulacral ridge, gonads, hepatic ceca. Okay. Now I'm going to show you some images. I'm going to clarify this. This is tends to be very confusing. So imagine I had on a top hat. And I took this top hat, or even a baseball cap or anything, and I took it off, and then I set it down on the table so you could see inside the cap. That's what we do. So what we've done here is we've removed the aboral surface, we've cut it off, and we're going to flip it down, and then we'll look internal. Okay? So just to reconstruct, you can just flip it back on, right? So just keep that in mind. That's exactly what you're going to see. Okay? So we're looking internal. Here's those digestive ceca. The gonads are underneath, so we don't really see it. This is the cardiac stomach here, because the pyloric stomach went with the cap that we took off effectively. So when we remove the aboral surface, we'll see that the pyloric stomach went with it. Okay? And then the ampullae are visible here. Now this is not reproductive season, so the gonads tended to be a lot smaller in this one. Here just shows you a close-up here. So we have here the cardiac stomach down here. Okay. Gonads, the digestive ceca are up more aborally, so they're not real visible, but they're back in here. You can barely see some dark shadow there. That's probably the digestive ceca there. This structure there is the stone canal. It would have led to the major porite on the cap, right? Remember, we took the cap and we lifted it away. So the major porite connects to the ring canal, which isn't visible. But the ring canal would be down in here, protected by the endosskeleton. Then it would lead out in the radius of each arm. So the stone canal connects the major porite to the ring canal here. Okay. Here it just shows you we clean this up a bit. So here's the cardiac stomach here. There's the gonads. Right there and there. Now, is this a male or female? It's hard to tell because the gonads are so small. What you could do if you had the opportunity is you could make a section of this and look to see if you see very small immature eggs or immature sperm. So that's how you do it biologically. But right now we can't tell with gross anatomy. Okay, they're just too small. Again, cardiac stomach here. There's that stone canal. And we can't see the major porite because it's been removed at that top. And the ring canal is within here. It's going to be protected. So when we have a dried starfish, you'll see the endoskeleton very clearly because the other tissue's dried away. Remember that this ring canal is around like this. The stone canal will have dried. And then we would see a radial canal here in this ambulacral ridge. Okay. Here's that inside of that cap effectively. Okay. So what we see here is what stomach is this then? Pyloric connected to the pyloric ceca. Okay? And I don't know if you can see like this brown-like structure sticking off of here. That's some of those rectal ceca. Because the intestine's so short we can't see it, but if you remember, this is a side view. You have to take this side view and flip it like this to get the same orientation. So we don't see the intestine, but we do see those large rectal ceca branching out from it. Those are the brown worm-like looking structures, okay? So we have pyloric stomach here, pyloric, hepatic, digestive ceca there, and here's some of those rectal ceca branching off because the intestine's behind here, we can't really see it, okay? All right. For the arthropods, this is by far the most diverse phylum in terms of number of species. There's well over 1.2 million described species now. The second most diverse phylum that we look at are the mollusks. They're about 100,000. Okay? So in terms of the cladogram, it goes like this. So arthropods here. Okay? And then we have the trilobitomorpha. These are the trilobites. They've been extinct for well over 200 million years. We do not have live trilobites, of course, in lab. Make sure you know the distinction extinct and extant. Extant is living. Extinct means that species is no longer present. It doesn't refer to an organism per se. It refers to that group of organisms. Okay? So in terms of the arthropods, we have the trilobitomorpha. Then we have two large groupings, the chelicerata and the mandibulata. These refer to specific mouth structures that they have. You've heard the term mandible before referring to the jaws. So mandibulata have jaws. And the jaws, in the case of the arthropods, go side to side in terms of fragmenting food, not up and down like we have, okay? So that's one group. The other group is Chelicerata. They have chelicera, which are modified feeding appendages, basically fangs that inject poison or venom. Those would be the spiders, ticks, scorpions, and horseshoe crabs. So the two groups that we have here in Chelicerata are the arachnids, the spiders, the ticks, the scorpions that I mentioned, and Xiphosaura, which are the horseshoe crab. And then with the Mandibulata, we have additional branching here. So you can remember the way the cladogram works. Chilopoda and Diplopoda are in the smallest group, Myriapoda, because that contains them. So if the question was, what's the smallest group that contains a horseshoe crab and a centipede, we'd have to go back here, and we don't give that term, so it'd be basically Arthropoda. Okay? I didn't give the branch name for there. So just Arthropoda would be that smallest group. Okay? So practice with that. All right? Myriapoda means many feet. We'll see that the Chilopida and the Diplopoda, these are the millipedes. They have 2 pairs of legs per segment, and there are many segments. And the Chilopoda have 1 pair per segment. So the centipedes, these are carnivorous. These are vegetarian, basically. And then this group here, I left this name off. And for those of you watching the lecture, there's a common quiz question, and that is, what's the smallest group that contains the Hexapoda and Crustacea? And that group should have the term Pan-Crustacea. Just take the term Crustacea and add PA in front of it. That's that grouping for the Hexapoda and Crustacea. Hexapoda include insects. Some people sometimes just call this insects. Insects are a specialized group within here. We just call them Hexapoda and Crustacea. And they're in the Pan-Crustacea. Okay? This diagram, there's some studies that show that students tend to understand the pattern of a cladogram better if it's written in this direction. So this just shows you that these 2 groups are in the Pancrustacea that I just mentioned, and these 2 groups are in the Myriapoda, and then the Pancrustacea, and Myriapoda, and Mandibulata, and Chelicerata, these groups, and then those are all with the arthropods. So if you find this way more useful for the other cladograms, draw them as boxes. Try that, okay? In terms of the arthropods, they have chitinous exoskeleton, so this is an exoskeleton, and that implies real limitations on how you get larger. So basically they molt, which is why these are in the group the ecdysozoans. For them to get larger, they will go through molting, okay? And they have lots of appendages, and they're jointed. And the joints allow them to have a lot of dexterity, fine movement, fine motor control, and to generate forces. And they're segmented, head, thorax, and abdomen. In a few cases, the head and thorax are fused, like in the arachnids or spiders, and that process is called tegmatization, the fusion of parts. We'll also see that in some crustaceans. Question? So the joints actually, you can think of it like the armor of like the old knights that would wear. The armor plates have a little gap between them, so that allows movement within them. Oh, so what creates the forces? They have to have attachment points within the endoskeleton. So let me just show you an image that will illustrate that more clearly. Here, we might illustrate it more clearly. So what happens is, so when we think about us, we can imagine we generate force because we can attach to the skeleton, so we get leverage points in that, right? So the way they generate forces here is they attach to the exoskeleton, so the exoskeleton is quite strong. So once they contract this, then that would, in this case, they shorten this, which would cause the leg to move midline, okay? Right? But they do have to have attachment points for that to generate those forces. And if you notice here, because they have multiple attachment points, it effectively allows you to rotate like this, that appendage, right? Okay? So let's go back to this. They still have external appendages, and they tend to modify them. And we'll see this really clearly in the crustaceans, where, again, with segmentation, they have it external as well. So now we're starting to see repetition of legs in that. And they modify those legs. In some cases, they might modify them in wings, etc. Okay, so here shows you molting. Now, we're going to have large chambers in the lab that we rear the cockroaches in. So you will do a cockroach dissection. Yes, there are gloves to wear. Okay, but please look in the cockroach chamber. And you're going to see maybe 100 cockroaches in there. You'll see some of them that are maybe potentially molting. So this one is crawling out of its old exoskeleton, and this is the fresh exoskeleton there. And what happens is it's reached its limit in terms of size. So it produces a new exoskeleton internal, and then it basically breaks out of its old exoskeleton. And once it emerges, in this case, it would suck in air, expand its body by the air, and then what happens is this exoskeleton, the chitinous molecules, the macromolecules that will oxidize and polymerize due to UV light. That's why it's a different color. And then that will harden it, and then basically they deflate their body, but now they have space to grow into. Okay? So that allows them to expand. And here we'll talk about, for the hexapoda, particularly insects, we'll talk about 2 different types of life cycles. Okay? We'll put this on the board. It's called holometabolous and hemi. So metabolism referring to metamorphosis. Holo is complete change, or indirect. And that's where you have egg, larval stage, a pupal stage, and then the adult. And they look very different. And oftentimes they exploit completely different environments, different resources. For example, like mosquitoes, they lay their eggs, fertilize eggs in water. The larvae are aquatic, they feed on algae, etc. in that water. And then they will go through this stage, this metamorphosis, and the adult will emerge. And so they exploit different resources there. Okay? Hemi is direct, so you don't see this huge change in body form. And so the larval forms aren't usually called larval, they're called juvenile, meaning they can't make viable gametes. Because they look like the adult, they'll sometimes call them nymphs or instars. We can see that on this image here. If you look here, this is a juvenile form. This is adult. This cannot make viable gametes. This one can. But they look very similar if you've seen this one already polymerized, okay? And then this just shows 2 of them side by side, where you can see they're about the same size, but this has not yet polymerized in that, okay? And then we already know this attachment point for generating forces. Here's the crayfish here. Here we see the head and thorax are fused together, and that's that example of tegmatization, okay? And here we see lots of segments here, and this is a male crayfish here. We'll go over this shortly again. If you look on the ventral surface, these have bilateral symmetry. So ventral surface, they have swim rats here that they use to swim with. These are walking legs here. So they use